I think we're ready. We can start. I'm happy to be here. So I think everyone almost knows me. So I'm part of the Bioimage Informatics facility, and I'm giving today's CBA seminar on next generation bioimage file formats in spatial biology. As a forward, it may sound a bit more boring than it hopefully will be. So we'll see. What am I actually going to talk about? So the goal is to look at, within the bioimage community, so only biological images, look at what's happening when it comes to file formats, data structures, and so on. And the target audience, so you should all be working with bioimage analysis to be interested there, but I think you all are. I'll do some introduction and also some terminology, and then I'll mostly discuss about bioimage formats. Yes? To me, it seems that all microphones here. That could be because I'm muted on Zoom. Thanks. Where? Here. That's good. No, it should work. Oh, thanks for monitoring. All right. Yeah, so I was saying that the goal for this presentation is to review ongoing efforts in the community to standardize data structures and bioimage file formats in the community, and that this talk is mostly aimed at bioimage analysts. And I'll mostly talk about bioimage file formats. I also, maybe if the time permits, touch upon file formats for storing tabular data derived from bioimages. As you all can see with all the tech set up here, so the seminar is being recorded, now hopefully with sound. And yes, please. Yes. 5. Rule number 5. They're strange. Yes. That better? Perfect. So yeah, the seminar is recorded and will probably be publicly shared. That's why all the tech is here. And now, of course, my pointer doesn't work anymore. All right. I start with acknowledgements. So I really want to, so I'm not, for the most part of it, I'm not presenting my own work, I'm presenting community work. So mostly work done by others. And this would not be possible without them really investing time and effort into this, partially in their free time. And what I would really acknowledge here is the work by Josh, who is behind a lot of these initiatives that I'm going to present. And also Joel for sharing some presentation with media that I'm using. Yeah, a little introduction. So if we go back to the title, it is Next Generation Bioimage File Formats in Spatial Biology. So I'll quickly talk you through the title. So bioimaging or biological imaging, I mean, it's clear any imaging technique that is used in biology. What many people don't really realize is that there are a lot of different techniques. They all work differently. Many are microscopy based, many are optical microscopy based. But there is also other techniques such as mass spectrometry based imaging, etc., etc. So the techniques that are used to acquire these images, they are really vast. So any file format that wants to unify this has to deal with a lot of different requirements. And what I also want to highlight is that these bioimaging techniques, I won't go through them all, they enable what we call spatial omics in parts, but when you talk about bioimaging, you usually don't include spatial omics as part of bioimaging. So bioimaging is a technique, and that is only part of spatial omics. So when I talk about spatial omics, this is, or spatial biology here, this is at its closest form, it's the study of biomolecules in any spatial context. So this can be biomedical or biological imaging. And again, there are many techniques, many of them very recently established, that in part use microscopy, but they can also rely on sequencing or other techniques. So microscopy is only a small part of this whole spatial omics thing. And again, even here, there's even a broader variety. So you measure either RNA protein, single cell to subcellular resolution. You can measure very limited multiplexity, so only tens of molecules up until to whole genome or transcriptome, depending on the technology, and many more variables. So a lot of variability there. And now, when you guys all come up with spatial data analysis for this, the tasks that you encounter, of course, you all know that, are very generic. So they apply to all of these methods many times. So except for pre-processing, which is usually modality specific, these common tasks such as spot detection when it comes to whole transcriptomic or proteomic approaches, or single cell data extraction, or then the whole spatial analysis, they are independent essentially of what the underlying technique is, the biological technique. So that's also something that file formats have to deal with. You want to unify all these techniques into one single standard that you can then apply many different spatial data analysis approaches to. And one big part that people often don't mention so much is visualization and quality control, which is a big part of that. And that's also a goal of file formats to enable that in a very easy way, so that you can use plenty of tools all on the same data without any hassle. And actually, Christoph from the facility is on Frederick. They're doing a really good job with tissue maps, I think. So yeah, all to say, this is often agnostic of the imaging technology. You see that basically from the segmentation part on, most streams that are outlined here, so that's different methods, they all follow the same core steps. And that brings me to computational infrastructure. So we've seen there are a lot of advances in bioimaging and then spatial omics techniques. A lot of these variables change. So you have multimodal imaging, 3D time, high multiplexity, etc. That all leads to larger and larger data. So really large data sets in the terabyte range sometimes. And not only that, it also leads to complex data. If you have like 50 markers, that might be manageable to visualize, but what do you do if you have whole transcriptome in space? How do you visualize that? And this in turn leads to very sophisticated analysis workflows that have their own requirements to file formats. That's basically what I list here explicitly. So novel methods that deal with this complexity, complex workflows, on the one hand are very generalizable, but on the other hand, they turn more and more resource intensive. So then you need things like cloud computing, clusters for running your computing. So that's where the file formats also need to be ready to support this. And that all leads to changing infrastructure requirements, both on the hardware side, so you need better hardware, better GPUs, etc., but also on the software side, and that's often forgotten. So, of course, well, the software infrastructure, a lot of things belong into or fall into that category, programming languages, obviously, but the whole software ecosystem as well, so do you have nice data structures, etc., but importantly, also standards. So that really you can rely on a standardized way of your data representation to speed up your use of algorithms, etc. And yeah, many more things fall into this software infrastructure, but what I'll be discussing here is file format. And file format, well, it's a standardized way of encoding any information. They have many different properties. They can be generalized, so general or more specialized. They can be layered. So, for example, one file format uses another file format in order to define a new file format, etc. And they are, in general, a structured way of representing data, but it's either chunk-based or directory-based. So, directory-based, you know, from your file system. That's basically a whole file system within a file. That's often used, but also chunk-based, so where the file is just split up into chunks and stored that way. And the purpose, yeah, data storage, access, and exchange, that's clear. But importantly, it is useless without any software ecosystem or standards. So if you cannot rely on finding that data specified in a certain way, then you are a bit at a loss. All right, so that was very general introduction, probably preaching to a choir that all knows this already. But in spatial biology, we have two essential data structures. One of them is images or raster images. The other one is tables. And I'll focus mostly on the images now. So by the way, this is from tissue maps, a nice example data set that Christoph probably has seen a million times now. Yeah, and I'll for the most part of it now, focus on the images. So when talking about images in spatial biology, you're mostly talking about n-dimensional pixel matrices. They contain a dimension order, so somehow these images are ordered in different dimensions, so spatial dimensions y, x, and z, and also t and c, so for time and then channel. They have a shape, so each of these dimensions have the size basically, and the data type. But importantly, they also have metadata, and this is what standards often try to focus on a lot. So for example, the individual channels that you have in an image, they have channel names. This would be considered metadata. And there are many, as you all know, proprietary file formats. Almost every instrument has its own format. And that, of course, is a burden to analysis. And that's why back in the days, actually it comes from somewhere else, but there was a file standard established, which is TIFF. You all know TIFF, you all work with TIFF. It was established in a time where fax machines were common, and that's why you'll often actually find multi-page TIFFs, because it's multi-page fax. But essentially what a TIFF file is a flexible and adaptable file format for any type of raster images. And they contain several subfiles or pages. So this is when it comes to multi-page. So for example, for different time points or C slices and so on. So each of them is a page. Each of them is what they call an image file directory. So I'm going through this because I think when you work with this every day, you might not realize, but it is important to understand what's coming after. But each of these subfile pages is called an image file directory that contains tags, and that where the name comes from, so the tagged image file format. And these tags can either be 2D image data, important 2D image data, with one or several samples per pixel, so that's the number of channels, so that can be RGB, or if the multiplexity is very high, that can also be 10,000s. And there are also metadata tags. So both are considered tags. And each of these IFDs can also contain sub-IFDs, which is very important. These are image trees. So this is when you talk about pyramidal images, then you have multiple resolutions, so that you can enable like a Google Maps-like exploration of the data. And yeah, there are many different features or properties of this file format. So it can be lossless, lossy, or no compression at all. And many other things that this format allows. And the problem with TIFF is essentially that, well, it has also been named thousands of incompatible file formats. It is so modular that there is no tool supports all possibilities that TIFF gives you. They try to establish what's called a baseline TIFF, but crucially for spatial biology, this does not allow high multiplexity. So that's either 3 or 4 channels, etc. So that's not really useful for us. And because back in the days, there was not even talk about terabytes, it's limited to 4 gigabytes. That's why a new format was born. It's called BigTIFF, which is technically not TIFF, but works like TIFF, just with different options. Yeah, TIFF standards, there are many of those. So many that actually individual tools have their own standard. ImageJ has an own standard. If you write using TIFF file in Python, you can set ImageJ to true, and then it will write somehow a TIFF file in a format that is understood by ImageJ but undocumented. So this is about what we are doing with every day. So some time ago. That was led by the open microscopy environment. So this is a consortium that tried to establish standards for bioimaging. They tried to come up with a more narrow specification of TIFF, which they called OME-TIFF. And this has had quite some success. What it essentially is, it's TIFF or big TIFF plus some defined metadata model. And this metadata is defined in XML, which is actually embedded in the TIFF header. So this is how OME-TIFF works. It's widely supported because it narrows down how a TIFF has to look like. So the tools don't have to support everything anymore. They can only support that subset of TIFF. However, the metadata is actually rarely used. So what this format achieved is you get an easy way to access your image data. But for example, ImageJ does not understand channel names that are saved in an OME-TIFF header. So the metadata is not really used. Nevertheless, it was quite successful. So it's quite good for small image file sizes. And it is actually the recommended format for the image data repository or LNCell Institute, among others. The big issue with OME-TIFF is scalability. So when you have really large data. And this is sort of the motivation what comes over the next slides. But OME-TIFF is, first of all, not really suitable for all instruments. It does not support parallel writing in different parts of the image, things like that. So it always requires some later conversion into TIFF or OME-TIFF. And that is obviously an overhead, especially for large data storage centers. But what is actually the biggest problem with scalability, or when it comes to scalability of TIFF, is that it does not support chunking. And what I mean by that is that the binary access to image data within an image is limited to these 2D tiles that I was mentioning before. So you can only load tiles, 2D tiles at once. Now, if you have a very large image, a 3D volume of light sheet data, and you want to get a small subvolume of in 3D, for example, what you will need to do is you need to load all the individual 2D slices, then crop them individually, and then reassemble them. Because you cannot, for example, have a tiled view in 3D. And what this also means is you can only have these image pyramids for different resolutions in 2D planes. So you can only have different resolutions for the individual 2D planes, but for example, not along the XC dimension, things like that. So this is really a problem for distributed object storage. So first of all, you have the chunking missing in the file, and it's all stored in one file. Okay, you can store the pages in different subtiffs, that's sometimes being done, but what all that means is not ready for distributed storage. And distributed storage, when it comes to large machine learning applications, is really important. So what that essentially enables is you store the data in many different places at the same time, and then you basically can scale your data access because you can use something. So first of all, you can use data locality. So if you need to have a small 3D volume, you ideally select whatever host is having most of the data you have. So if you're distributed and then repeat it along many hosts, that's relatively straightforward then. And second, of course, it's all about concurrency. So if you can split your data in small parts across different places, that's a good thing. And finally, yes, it's highly specified, but actually the metadata of OME-TIFF cannot be extended at all. Imagine a viewer wanting to save metadata on how the image was last viewed, what camera orientation, etc., cannot be put into these images. So that's why so Josh Moore came up sometime, he's part of the OME consortium, came up with what's called OME NGFF. NGFF for next generation file format. He chose the name as a working title, it stuck around. So yeah, you can all have your own opinion about the name, but it was essentially published first on bioRxiv extensively. It's an interesting paper to read, but also highlighted then in Nature Methods and other places. It is chunked, so it is ready for cloud or object storage. It is pyramidal, agnostic of the dimension you look at, so it does not have to be 2D slices. And importantly, it is community driven. So there is no benevolent dictator for life that runs the whole project. It is really like everybody can contribute to it with all its up and downsides to it. It is a specification only. There is per se no implementation of it. So it is really only a specification that determines the logical structure how this file should be organized. And it has different storage backends. So you can use OME and GFF using HDF5 if you know HDF5 or other. So you can apply this specification of a hierarchical structure to multiple different underlying file formats, general purpose file formats. But there is sort of a reference implementation. There is currently no one really paid to develop any reference implementation, but this was just the first implementation that came up, and this OME SAR is also used as part of documentation in the examples. Yeah, here's just an illustration on the pyramidal images. Now, when I say reference, there's a reference implementation, I'm mostly talking about this OME SAR. And this is OME and GFF using the SAR file format. And before I can talk about SAR, I need to quickly talk about data storage. So data storage can happen in many different ways. For example, block storage. You chunk up your data in equally sized blocks and store it, for example, on a hard drive. So hard drives are the definition of block storage. File storage is, you'll work with that. All the data is stored in a single file, basically, that is self-contained and part of a hierarchical structure. But then there's object storage. So I mentioned that before. That is a way, for those of you that don't know that, to store data in a distributed fashion in the cloud, basically. So it's made as a cloud format. And here, data is split into separate self-contained units. So each of these units has a meaning. So for example, I define this unit is that part of an image. Unlike, for example, block storage, where it's in equally sized chunks without any meaning what these chunks are. And these self-contained units can then be distributed in the cloud or wherever, or even locally. And now SAR is one of these file formats that for chunk compressed n-dimensional arrays. So that's a very general definition. So you can store your NumPy arrays, whatever, in SAR in a chunked way. And in SAR, every chunk, it actually corresponds to a file on disk. So this is where it gets a bit confusing. So we go back basically from object to file, but every chunk that you have in an image is stored as a separate file on disk. And now NGFF is a specification that describes the hierarchical layout of these chunks, how they are stored in what hierarchy. And now if you apply that, you get OME. And this is here on the left. So if you have, for example, TIFF here, you would have a 120 gigabyte image. In SAR, this would actually be what an OME SAR file actually is a directory on disk with many subdirectories that each contain the different chunks of an image. What this allows is high performance access of regions of an image. So this is from the original publication of OME SAR, where you have the individual rows for SAR, TIFF, and HDF5, all of, so that's image data, simulated image data, all of different, of similar size, so 86 gigabytes. And now if you start with TIFF, you see if you want chunked access of an arbitrary region, of course, because of all these loading of individual layers, you are quite in a quite high machine here on overall data access. Whereas if you then compare that to SAR, of course, you're lower because you can really fine-grain your chunking strategy and access the chunks in a performant way. HDF5, by the way, is a similar format to SAR. only that it has the disadvantage that it does not allow parallel writing. So in SAR, because it's individual files rather than one big file, you can write the individual files in parallel, whereas with HDF5 that does not work. So this was part of the original publication. It's gotten better since then. But what this essentially enables, so this is a video from Michel Muti that I mentioned before, is basically in a Google Maps way, zoom into a, so this is a, I think, 150 gigabyte image or so, and on the fly load this from cloud storage. So this is stored somewhere on the cloud, and he just on his laptop browses this 150 gigabyte image, which is quite impressive. This was, I think, high content screening plate. So these are 23 wells in a high content screening plate. And I'm actually working currently on a similar data set, and it's been really, really useful so far. So yeah, as I said, standards don't help without tooling. So there is a lot of work being put into developing readers, writers, file converters, image viewers, and even whole platforms that rely on this standard. If you want, you can check out this link. Part of a hackathon here, I evaluated some of these, how good they are and bad they are, but essentially what remains is a lot of work still needs to be put in. This is a community effort. So if you ever feel like contributing, please do. Image viewer, actually we are trying to, so we are trying to adapt tissue maps in a way to load this data via spatial data route. I may have the time to talk about that. But what I also find impressive is that whole institutes are built upon this format. So not institutes, whole facilities are implemented upon this format. So there's a lot of hope for this to become a standard in bioimaging for all modalities. Yeah. There is quite some work to be done still. So on that here I only list some things that I am excited about when it comes to the specification. One of them is sharding. That's actually not part of OME and GFF, but from SAR. So the upstream, the part that is used by OME and GFF, which actually goes back and stores multiple chunks in a single file. Because if you use OME and GFF locally, you end up with 100,000 files, maybe. That's not efficient with current file systems. With cloud storage, it is. With current file systems, it isn't. And this is sort of what sharding addresses. You can combine chunks again into individual files. And part of the OME specification, there's also quite some work being put into the new release. So currently, the specification has been version 0.4, 0.5, I think. So for the next one, what will come definitely is coordinate transforms. So for example, if you have this high content screening where you have individual wells in space, you also want to describe how they relate to each other. How they relate to each other in space. So where they are relative to each other. So this format also allows to store coordinate transforms that do this description. And there is an improved version of that. What's crucial, I think, is it's currently being figured out how the project should actually be run. It's actually the first bioimage file standard that is community driven. It's everything else before somebody came and said, this is how we do it, with some more or less consultation. But this is meant to everybody just writes pull requests, essentially. It's a GitHub repository, everybody contributes to it. And what currently in the works is an RFC-like process, so the IETF has an RFC process to propose new features, etc. And this is currently in the works. But also, what's still missing is namespaces, which essentially allow extensions. So if I'm the developer of tissue maps, I can then say, tissue maps needs to store this data in order to view this image. And I can write this using a tissue maps namespace as an extension to the standard. Yeah. So if you feel like it, please get involved. I don't know how are we doing time-wise. I think we can. Yeah, I'll briefly talk about tables. Why tables? Well, anything we extract from images essentially ends up in tables. So could be single cell data, where you basically have a two-dimensional matrix where the rows are cells, the columns are features of the cell. So that's why it is important to see this in combination with imaging. Ideally, you want to store this together. Well, often stored as common separate file, that's clear. But in spatial biology, we rely a lot on metadata, channel names, etc. So this is not really possible with CSV. We also often want layers. We want to transform our data, argc transform, and then write it to disk. Then that's a second layer to the original data. And usually the data is very sparse, so you can compress a lot. And there are a few Python packages I'm probably preaching to the choir. You've mostly used them, I guess. One of them is undata, which is a Python package for annotated data matrices that is placed somewhere, for those of you who know, between pandas, so the tabular package, the tabular package in Python, and Xarray, which is very general, which can store n-dimensional matrices. And undata is somewhere in between, so it can store multiple layers of like tabular data with both row-wise and column-wise observations that in turn can again be either individual values or actually n-dimensional. So this has a custom file format based on HDF5, so very similar to SAR. And is used by pretty much all Python-based single-cell analysis packages. There's an extension to do multimodal data, but mostly it's used. On the R side, I won't discuss this much, but on the R side, there are two very important data structures as well, single-cell experiment and spatial experiment. And on the R side, this has actually achieved what file formats are trying to establish. So everything uses this except zerout, which is quite important, but everything else in R/bioconductor uses one of these two data structures. It's not yet the case in the Python world. But the reason I'm mentioning this is spatial data. What spatial data is, you can think of it as an extension to OME and GFF. So it can store images. But on top of that, it supports all major data types that exist otherwise. So not only images, but also tables, for example. And there it actually reuses undata. So it can store tables, it can also store labels and regions, etc. And this is sort of seen as the prototype for potential new developments in the NGFF space. So a lot of what the spatial data subcommunity is trying to define will eventually maybe end up in OME and GFF. So this is really exciting because for the first time you can actually store individual modalities, so individual data types within a single file, which means that basically if you say now I want from this subcrop of your volumetric image or whatever, I want to get all the points and the crop in the image and all the regions in that, it's all co-registered. You don't need to deal with different types of files, different ways coordinates are stored, etc. So this is actually really exciting. What else should I say about this? Yeah, so again, spatial relations are stored via transforms, so it is made for spatial omics. It aims to support all existing spatial omics techniques. And they say it's language independent. However, most of these underlying things like SAR, implementations for them mostly exist in Python. So the standard is language independent, but if you want to use it, currently you will need to use Python. Yeah, and it already has quite some nice integrations, including PyTorch and also a guy forgot to mention before, but the SAR community is also actually very closely working with other types, so not just imaging, but also Google, for example, has an extension for TensorFlow to deal with SAR data. So that all integrates nicely together. Yeah. So this is actually what I wanted to talk about today. I hope there was at least something new for you in there. But the key message I think that I want to leave here is that this software infrastructure is an essential part, as you all know. Therefore, yeah, it's good to acknowledge the work at least and ideally contribute to this work as well. Yes. And that's